So the nucleosome is 10 to 11 in nanometer in its diameter. You can see the mention. I have mentioned the dimension here. 10 to 11 should be the diameter of the nucleosome. And nucleosome is made up of octamer of histone. Octamer carries H2 alpha 2, H2 beta 2, H3 also 2, and H4 also 2. So two molecules each. So 4 types are there. 8 means octamer. So histone octamer forms the new body or the core, and their positively charged groups are towards the outside. And DNA being negatively charged, its negative charge portion is the phosphate group. So they interact with each other to stabilize the DNA, and it is not 2 turns of DNA around it, 3/4 turns of the DNA around it to form a nucleosome, which is 10 to 11 nanometer in its diameter. Now DNA connecting the two adjacent nucleosome is called as the interbead or the linker DNA. Now you need to understand this point, that nucleosome, when they are formed, this is one nucleosome, another nucleosome, another nucleosome. So nucleosomes are joined with the help of the linker DNA. So the piece of DNA around this will become a part of nucleosome, and the piece of DNA which joins two nucleosomes together is called as the linker DNA, or also called as interbead, because each nucleosome looks like a bead. So interbead or linker DNA. And remember, this DNA will carry the histone 1. Histone 1 is the linker histone, and remember this one is the remember this point here, that all these proteins H2A, H2B, H3, H4, they all are one of the most conserved proteins. So please remember, H1 is the most non-conserved protein. So H1 linker is present in the linker DNA. So it bears the H1. H1 is not conserved but is quite specific for different tissues. A typical nucleosome contains 200 base pairs. Now you need to understand this also. The DNA turns around the octamer. Now how much DNA turns around? It is 200 base pairs according to NCERT, but to be more precise, it is 146 base pairs around, and then 54 base pairs, total becomes 200. 54 base pairs forms the linker portion. So linker is made up of 54 base pairs length of DNA, and the DNA which takes a turn around to 3/4 turns around the core of the nucleosome is 146 base pairs. I'm repeating again, a typical nucleosome has 200 base pairs, but reality is 146 base pairs will take a turn around, and then 54 base pairs will form the linker DNA. So a typical nucleosome has 200 base pairs, and remember these two portions join to form a chromatosome. So nucleosomes, when joined to the linker DNA and the linker histone, this forms a structure which is called as chromatosome. So a large number of chromatosomes join to form a chromatin. Chromatin word you have already done even in the 11th class. So now the next point. A typical nucleosome contains 200 base pairs of the DNA helix. When viewed under electron microscope, nucleosomes in chromatins are seen as beads on the string. So beads are nucleosome, string portion is the linker portion, and each bead with its string is called as chromatosome. Beads will carry 146 base pairs of the length of DNA. String will carry 54 base pair length of the DNA. Together they will form the chromatosome. I hope this point is clear. A nucleosome is the first level of shortening of the very long strand of DNA. Now imagine it's a long thread of DNA. So when it starts coiling around, the histone, a great length of DNA, a great portion of the length of DNA is used up. So that's why it starts becoming shorter in length, because you are wrapping it around the spherical structure. So nucleosome is the first level of shortening of a very long strand of DNA. Nucleosome constitutes the repeating unit of a structure of the nucleus called as chromatin. Chromatin is nothing but a thread-like stained body which is seen in the nucleus. A typical nucleosome has 200 base pairs. DNA of a typical mammalian cell is this much long. So how many number of nucleosomes or beads on a string will be there? You can perform this calculation. 3.3 10 raised to the power 7 nucleosomes are present in each mammalian, or you can say human cell. Beads on the string structure of the chromatin coils and further coils and undergoes condensation to form what is visible during metaphase, and you call them as chromosomes. This packaging at the higher level requires additional set of the proteins which are called as non-histone proteins or non-histone chromosomal proteins. Now let's understand how this packaging takes place. This is the DNA we have already learned, and the size of the DNA is 2 nanometer. Now this DNA starts coiling around the core. As it starts coiling around, a great length of DNA is already used up to coil around, and then this is the linker, again coil around, linker, again coil around. It goes on like this. Now the 2 nanometer thing, which is very much long in size, has become 10 nanometer thick. So the thickness is increasing. Just remember, as the thickness of this molecule increases, its length is going to decrease. So that means this is what happened in the metaphase also. If you remember, at the metaphase stage, the chromosomes are thickest and the shortest in size. So DNA is quite thin, 2 nanometer, and it is very much long, but as it starts undergoing condensation and packaging and supercoiling and further coiling, it will become thicker and shorter in size. So now from 2 nanometer, the thickness or diameter has become 10 nanometer.